So the next group of fish I want to talk to you about are the Cyprinidae, which are the minnows. Now this is a huge family. They're worldwide, and I'm going to start by talking about our native minnows, and then later on we're going to talk about the non-native minnows, which are kind of a big problem here in the US. Our native minnows are all pretty small fish. They're very interesting. There are about 72 or so species of minnow in Kentucky, and they all are very unique. So don't just call a small fish a minnow. A lot of people will do that. A lot of people lump all these little small fish, just call them all minnows and treat them all the same. Don't do that. They're all very interesting. They have names. They are important to the aquatic ecosystem. They are a big part of the aquatic ecosystem. They're maybe not as important to a lot of people, but they are certainly important to a lot of the fish that people go after. So they're important prey for larger fish. They're important water quality and habitat indicators. And so if you don't have good water quality, you won't have a lot of minnows, and so that's a good way to check for the water quality. They're mostly found in streams. You will get some in lakes and in still water bodies, but most of these species are in running water. They're great for aquaria. They take prepared feed well, they survive well, they're active, they're interesting, so I would recommend them for tank fish. And I think I already mentioned this, but we'll mention it a little bit more. They are essential links in the aquatic food web. You've got herbivorous fish that will eat algae and some plant material. Also, you've got sometimes detritus. A lot of them are insectivores, and they eat other invertebrates. There are some carnivores that eat larger items and maybe even some smaller fish. So this is how the energy flows from the primary producers to those larger fish. And that's another reason why this group is very important. So one of the ways that we identify these fish, and one of the first things you find in the keys, we're sort of looking at carnivores versus herbivores. And we use this for identification because of their internal anatomy. The carnivores tend to have a silvery peritoneal lining and an S-shaped stomach, and they actually have a stomach. Whereas the herbivorous fish tend to have a more black or dark lining to the coelomic cavity, and they have a very long coiled intestine and usually don't even have a stomach. So they need that herbivorous material to stay in their intestines for a long time to get broken down. And so here's a couple of pictures that show you the differences between those two. And you can see the S-shaped stomach versus the coiled intestines of the herbivore. Here's another silvery peritoneal cavity. Here's another dark peritoneal cavity. Sometimes people have a question, is this silver, is dark? Look at these pictures, it's pretty obvious. And again, almost always the dark cavity will have the long coiled intestine, and the silvery cavity will have the S-shaped intestine. So you can use those to kind of work through your key. So how are we going to recognize these fish in general here in Kentucky? Like I said, they're small. They've got cycloid scales. The two main characteristics are that they only have a single dorsal fin and they have a forked tail. And so that will help you separate them out from a lot of the other smaller fish families we have in Kentucky. Also, during the breeding season, the males get these breeding tubercles. And the catastomids also get those. Again, the catastomidae, which are the sucker fish, evolved from the Cyprinidae. So they share a lot of characteristics. So here we've got two common small fish that we're going to get, but in the top picture we see we've only got a single dorsal fin, whereas the darter, which is in the perch family, different family, has two dorsal fins. Also, the minnow has the forked tail, whereas the darter has a more truncate tail. So if you're looking at a small fish, this is one way to identify the Cyprinidae family. Here's an example of a male with some breeding tubercles. These are hard, bony projections, usually on the head, sometimes on the fins, and the rest of the body. So identifying these species is not as easy as with some of the species we've talked about. We've been talking about things like gars and bowfins and paddlefish. Those are all very easy to identify. Now, we're going to start earning our paycheck. The minnows are small, so you almost always have to use a dissecting scope to see these. You got to get used to that. You need to take your time. And the first few times you work through the key, it can be a little frustrating, but you will start to recognize certain characteristics pretty quickly. And the key that we use, the Fish of the Tennessee, is pretty good. And if you follow it, it'll help you identify these fish. You're going to need to cut them up. They're dead. You can't hurt them. You can't avoid cutting into these fish to look at some of the internal structures. Some of the common structures we use are the pharyngeal teeth. These can be used to get your final identification down to species. And so here are some examples of pharyngeal teeth that you'll find in minnows. And the way these are arranged right now is that you're looking down the throat of the fish. Now, in the key, and by convention, these teeth are numbered by the number of teeth in each row, and the rows go from left to right as you're looking down their throat. And so you start with the outer row on the left, then the inner row on the left, the inner row on the right, and then the outer row on the right as you're looking down the fish's throat. And so then the key often indicates how many pharyngeal teeth are in each of those rows. So for example, this species is a 2-4-4-2. So if you look down the throat, which is what we're doing in this picture, you've got 2 teeth, then 4 teeth, then 4 teeth, then 2 teeth. All right? And they're usually symmetrical, but not always. Now if you look at this example, these 2 horns at the top, those are not teeth. That's where the pharyngeal arch attached to the dorsal part of the opercular chamber. All right? Those are not pharyngeal teeth. This first row, you've got these 2 small pharyngeal teeth. And then in each of the middle rows, they're showing 4 teeth, and then 2 on the outside of the other pharyngeal arch. Now, those small ones on the outside arches, or on the outside rows, are often difficult to see. They're often delicate, and they break off easy when you're trying to remove them. So you got to be careful when you remove these arches, and you need to watch for that. A lot of times if you break off a tooth, you can see a little nub where you broke it off, and you can kind of tell that you did break off the tooth. So you just have to be careful, but this is something you're just going to have to do to properly identify these minnows. Now I made a video talking more about pharyngeal teeth and showing exactly how I remove them and clean them up and use them. So I posted that link on the Canvas page. Watch that, and it shows you how to get at these teeth. Another thing that we use a lot in the minnows is counting fin rays. And I talked about this, how by convention the way we count these is a little bit different in the Cyprinidae. As with any fish, to count the fin rays, you need to spread that fin out and light it from behind. And that really helps you see the fin rays. And also it helps sometimes to take a probe and sort of feel along the fin and count the rays as you tick along with that probe. And so here's an example. Again, we're lighting that fin from behind and we're stretching it out. Almost always need to use a dissecting scope. It really does help a lot to pick these things out one by one if you're under a dissecting scope. Now, I'm showing here with the probe, I'm showing you in this picture the small, most anterior ray on the dorsal fin, and it's usually small and tightly bound. And in the Cyprinidae and the Catastomidae, we do not count this one. We only count what are known as the principal rays, which are the larger rays. Now, in other families, you count all the rays. They don't have this little short one in the front. But in the suckers and in the minnows, they often have this little short one, which we don't count. And like I said, we count the principal rays, which are the larger rays. And usually the first principal ray is unbranched and the rest of them are branched. That's what we count when we're working through the key. And often at the posterior edge of the dorsal fin, you're going to have two rays that appear to connect underneath the skin. And if they appear to connect underneath the skin, they probably do connect underneath the skin. And so by convention, those are counted as one. If it looks like they join beneath the surface of the skin, which is sort of what I'm showing you in this picture. To me, in this picture, those last two rays look like they join just under the skin. So we would count that as one, not two. All right? And so that's just an introduction to the Cyprinid family. And in the next part of this one, I'm going to show you some specific examples that we get here in Kentucky, and I'm going to try to point out some of the characteristics we use to identify those species. So I'll see you in the next part.